Welcome to the SAN Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we dive into today's topic, if you enjoy what we do here and find these deep dives valuable, please take a moment to like this episode, subscribe to our channel, and share it with your friends and family. Your support really helps the SAN Science Experience grow and allows us to keep bringing you cutting-edge research. Today, we're plunging into a really fascinating discovery from a recent Nature Aging paper. It pulls back the curtain on a fundamental yet previously sort of hidden mechanism within our very cells. We're talking about the powerhouses of life, mitochondria, and a newly identified system that appears to be absolutely crucial for keeping them healthy, efficient, and maybe even extending lifespan. This isn't just about cellular mechanics. It feels like a foundational piece of the aging puzzle. Okay, let's unpack this.
Indeed. It's quite exciting. This work from Yanbai, Chonglin Yang, and their team introduces us to something they've called Mitochondria-associated translation organelles, or MATOs for short. You can sort of picture them as these tiny, incredibly dynamic molecular factories. They're not just near the mitochondria, they're intimately associated with their surface, playing this vital on-site role in keeping them running properly.
Right. MATOs. So our mission today is to go beyond just, defining them. We want to understand how they operate, the impact this discovery has on cell biology, and critically, what it could mean for tackling aging and disease down the line. It's like a detective mission into cellular longevity. So maybe let's start with the basics for a moment. Most of us probably remember mitochondria as the powerhouses of the cell, right? They make ATP, the energy currency.
That's the common analogy, yes. And it's true. They generate ATP, which fuels almost everything. But their role is much more complex than just being a simple power plant.
Yeah.
They're intricate structures, double membranes. The inner membrane folds into these things called cristae. And those folds aren't random. They massively increase the surface area needed for, well, a whole cascade of processes. Things like the tricarboxylic acid cycle, the electron transport chain, oxidative phosphorylation. The core of cellular respiration happens there.
Okay.
And when there are defects, not just in function, but in the structure, in those cristae, the consequences can be severe. Think serious neurological and developmental diseases. What makes it a biological puzzle, though, is that mitochondria have their own tiny bit of DNA, but most of the proteins they need, thousands of them, are actually encoded in the cell nucleus.
Right, I remember that. So they have to be made elsewhere and then brought in.
Exactly. Synthesized in the cytoplasm and then imported very precisely into the right mitochondrial compartment. It's a huge logistical challenge for the cell.
And this is where the cell gets clever, organizationally speaking, and where this paper adds a big piece to that puzzle. How does the cell manage that traffic so precisely? The study brings up these things called membraneless organelles, MLOs, which sound different. Not like the usual organelles.
That's right, no lipid membrane. Instead, they're dynamic, self-organizing compartments. Cells create them through this fascinating process called liquid phase separation, LOPS.
Liquid phase separation.
Oh, okay. Think of it like oil and water separating, but inside the cell at a molecular level. Specific proteins, often ones with these intrinsically disordered regions, kind of coalesce into dynamic condensates.
Like little temporary workshops.
Exactly. Workshops that pop up right where needed, bringing the tools and workers together without needing walls. Before this work, honestly, their specific role in mitochondrial health or homeostasis was surprisingly unclear. This paper really shines a light there.
And the key player, the sort of foreman in this mitochondrial workshop, is a protein called LARP1, law-related protein 1.
Precisely. They identified LARP1 as a key RNA-binding protein, an RBP, that's central to organizing these workshops, specifically for mitochondrial maintenance. It's like the conductor bringing everything together.
Okay, so LARP1 is the conductor. This leads us to the core discovery in C. elegans, the worms they studied. This is where it gets really interesting.
It really does. They found LARP1 orchestrates the gathering, the coalescence of the translation machinery, so ribosomes, the protein-building machines, along with other RNA-binding proteins. And this happens via that liquid phase separation process, forming these distinct MATOs, the mitochondria-associated translation organelles.
And the associated part is key, right? They stick to the mitochondria.
Absolutely key. They uniquely and intimately associate directly with the mitochondria. It's a direct physical partnership.
So what's their job right there on the surface?
Their function is critical. They mediate the local synthesis of specific proteins right there on the mitochondrial surface. It's incredibly efficient.
Instead of making them far away and shipping them in.
Exactly. MATOs ensure crucial mitochondrial components are made precisely where and when needed. The paper gives great examples. IMMT1, which helps shape those vital Cristae folds, and ATP2, a subunit of ATP synthase, essential for energy production. Both are synthesized right inside these LRP1 MATOs.
Wow. So it's like just-in-time manufacturing for the mitochondria.
That's a perfect way to put it. Immediate production, immediate delivery. It bypasses potential traffic jams in the usual protein import pathway. It's a shift from central manufacturing to a distributed system.
That sounds absolutely vital. So if it's that important, what happens when it breaks down? What did they see in worms without functional LRP1?
They looked closely at that, and the effects were quite dramatic, really hammered home how important MATOs are. When LRP1 was gone, the mitochondrial defects were severe. They became abnormal shapes, kind of spherical, bloated, losing that healthy elongated form.
Okay, structurally compromised.
And internally, the Cristae organization was profoundly impaired. Those crucial folds were messed up, and the protein levels, they plummeted. A staggering 84.5% of identified mitochondrial proteins were downregulated.
84.5%, that's almost everything.
It's a near total collapse of the building blocks. And function followed structure. Significantly lower oxygen consumption, so lower metabolism, and drastically reduced ATP production. Their powerhouses were failing.
And did that affect the whole worm?
Oh, absolutely. These cellular problems translated directly to the organism level. Fewer offspring, higher rates of embryos dying, slower development.
So problems growing and reproducing.
Right. And crucially, a significantly shortened adult lifespan. It's clear evidence. MATOs, orchestrated by LRP1, are essential not just for cell health, but for the whole organism's vitality and longevity.
That really paints a picture, but I'm still wondering, these are dynamic, membraneless droplets. How do they actually stay attached to the mitochondria? What's the molecular Velcro here?
Good question. They investigated that too. They found MATOs associate with mitochondria in a way that depends on the TOM complex, translocase of outer membrane.
The TOM complex, that's the main entry gate for proteins into the mitochondria, isn't it?
Exactly. The primary gateway for most nuclear-encoded proteins. And what they found was fascinating. If they used RNAi to reduce levels of TOM components, TOM20, NETA22, NETA40, NETA70, they saw MATO numbers drop severely.
Like cutting the anchor rope.
Pretty much. And mitochondrial health suffered concurrently. But here's the elegant part. The interaction is direct. LRP1 physically interacts with TOM subunits, and even more, these TOM subunits actually get sorted into the Electron condensates. They become part of the MATO itself.
Wow, so the gate becomes part of the factory.
In a way, yes. It provides this clear mechanism for coupling local protein synthesis directly with immediate, efficient import. It's a beautifully coordinated system. Ensures the parts get made and installed right away.
That's incredibly sophisticated. All right, let's shift gears slightly to maybe the most compelling part. The link to aging and stress. What happens to these MATOs as the worms get older or stressed out? Does this system decline?
Yes, and the findings there are quite revealing. In C. elegans, MATO formation naturally goes down with age. They peaked in early adulthood, maybe day 1 or 2 after the final larval stage, but by day 5 they were hard to find.
And that decline matched mitochondrial problems.
Directly correlated. As MATOs disappeared, they saw more mitochondrial fragmentation and dysfunction. Same thing with stress, like starvation MATOs decrease rapidly within hours. So it looks like MATO loss is part of normal aging and a response to stress, both contributing to that age-related decline in mitochondrial health we see.
Okay, MATO decline equals mitochondrial decline equals aging, but then they did something really clever, didn't they? This mitochondrian persistent MATOs idea.
Ah, yes. This was the truly groundbreaking intervention. They used genetic engineering to essentially tether LARP1 directly to the mitochondrial surface using GFP. They basically forced the MATOs to stick around, even in older worms or stressed worms.
They stopped them from disappearing. What happened then?
The results were, well, frankly remarkable. These persistent MATOs worked incredibly well. They protected mitochondrial health, delayed fragmentation and dysfunction during both aging and starvation. Animals with these anchored MATOs kept much higher ATP levels and oxygen consumption rates later in life. Their energy production stayed robust for longer.
Okay, that's significant. Better mitochondrial health for longer. But the big question, lifespan.
And here's the showstopper. These anchored MATOs massively extended the adult lifespan of the worms by over 43%.
43%, that's huge.
It's a profound difference. Imagine adding nearly half to a human lifespan, relatively speaking. And what's really significant is how broad this effect was. It worked even in worms that were already genetically programmed to be short-lived, like DAF16 or HSF1 mutants.
Wow.
And incredibly, it further extended the lifespan of mutants that were already long-lived, like ACT1 or ACT2.
So it wasn't just overriding one specific pathway.
Exactly. Its broad impact across these different genetic backgrounds strongly suggests MATOs tap into a really fundamental conserved mechanism influencing longevity. It implies we found a key lever in the aging machine itself.
So summing this up, it really feels like we've uncovered this crucial, probably evolutionarily conserved mechanism. LRP1 orchestrating MATOs is essential for keeping mitochondria working, which directly impacts health and how long an organism lives.
It absolutely is. It's a real aha moment.
Yeah.
Local protein synthesis in these dynamic, membraneless organelles is a core strategy cells use. And knowing this opens up completely new ways to think about therapies. Think about diseases linked to mitochondrial problems, Parkinson's, Alzheimer's, metabolic disorders, maybe even just general age-related decline. It definitely raises the question, could we find ways to boost MATO function or persistence in humans? Could that be a viable strategy to fight these diseases, maybe even promote healthier longevity? The potential feels enormous.
It really does. Well, that's it for our deep dive today into the really fascinating world of mitochondria-associated translation organelles. Incredible stuff. We hope you've enjoyed exploring this cutting-edge science with us and that it sparked your curiosity about how our cells actually work. And remember, if this deep dive into MATOs got you thinking, please show your support for the Sans Science Experience. Like this episode, subscribe to our channel, maybe share it with someone else curious about health and aging.
Imagine the possibilities if we could harness these tiny cellular factories to keep our mitochondria humming for longer. What could that mean? Until next time, this has been the Sans Science Experience.